So today we are going to see the anatomy of the Dicot root. So firstly we have to cut a transverse section with a blade. After that we have to dip it in the coloring agent. After that we have to put it in the slide and then cover it. Be careful while covering it so that the air bubbles should not be there while seeing it in the microscope.
Morning everyone, I Aditi Sharma is here to do the experiment on monocot stem of a monocot plant. This is a monocot plant. Its stems, its leaves are of parallel venation. So first we can cut its transverse section. Then we will take it off and put in saffron water. After dipping it in saffron water, we will put it on slide and cover it with a cover slip. Slide is prepared of monocot plant stem.
Good morning everyone. Today I am Dicot standing in front of you to perform the dissection of monocot root. In this dissection we are going to understand the basic anatomical feature of monocot root. First of all, we will remove all the root hairs so that we can observe its efficiency. Now we will put it in the coloring agent.